vaccinations, an update on COVID-19 vaccinations. So we have with us my parents, Chayaji, Ashish, Seema, and so I'll first just hand over to them and allow them to just quickly tell us about themselves, how they are, and what their expectations from today's talk, and then we will start the actual talk in about 3 to 4 minutes. Okay, Daddy, Mummy, over to you. Please unmute yourself first.
Yeah. Well, in India, it's okay, under control, and the vaccination is going on at a fast pace. However, my anxiety is that this Covishield of Oxford University in Europe, in some of the European countries, they say it should not be inoculated to people above 55 years of age or 65 of years age. And the second is our own COVAX, Bharat Biotech vaccine. It has not declared anything about its efficacy, effectiveness. So again, that is also under question whether it should be taken or not. So I would be happy if that can be clarified whether this should be taken or we should wait for more time.
Thank you so much, Dr. Sapan. God bless you and everyone. And I'm ready whenever we'll get ready to take vaccine. And thank you so much. Over to God bless.
Hello, my name is Chaya, and I've been attending these talks for a while. Very informative and raises a lot of questions. So today's vaccine, when we going to get it? So, and my question will be: would we know which vaccine we will be receiving when we get one? So my question will be that. So, we can sort of look, because every country is getting bunch of vaccines from all over the place, mix and match and all that. So if the first vaccine we get and that stock is not available, will they give us a second one from Moderna or something else? So that's my question is. Thank you.
Hi, everybody. My name is Ashish Kothiyal, and I'm from Toronto. I have been attending all of Dr. Sapan's online sessions as well as his blogs. They are very informative. And in today's talk, I would like Dr. Sapan to shed some light on whether these vaccinations would be effective against various strains. I was reading on internet and through various news, there are more than 4,000 strains now around the world. Would these be effective against all those strains? Thank you. Over to you, Seema ji.
Hello, everybody. I'm Seema Nair. I'm a registered holistic nutritionist. As a holistic nutritionist, I would like to ask Sapan about all the long-term side effects of the vaccines. That is what I am interested in. The long-term side effects. Yeah. Especially in different types of age categories, starting from children's, adults, and seniors. So what would be the long-term side effects? That is what I would like to know more. Thank you.
Thank you, everyone. That was a fantastic little introduction. So those are some really, really good questions. So parents asking about efficacy of Bharat Biotech, and also the question about countries like Germany and Norway, I believe, who have decided to not license the AstraZeneca vaccine above the age of 65. Very important question. Ashish's question about strains is very important indeed. Chayaji's question about availability of vaccines, and Seema's question about side effect profile. I mean, we are really covering everything with those questions. So Ashish, I need one more minute to just check my stream health. So Ashish, can I just hand over to you to just talk about the strains a bit yourself and what information you have about and what your concerns are about strains?
Sure. There's a lot of information available coming from various sources, coming from different bodies, and everybody has their own opinions. Somebody like uh, Mr. Rawat said that some are saying Covishield should be given above 65. Some are saying, some countries are saying no, it should not be. But of course, we have been waiting. We are in a wait. We don't know when we will get vaccinated. But right now, there are so many. I was reading there are more than 4,000 mutations that have happened around the world. UK is one, South Africa is another one. They are saying in every pocket around this world, the virus continues to mutate, and it's a I think I would like to hear from experts or Dr. Rawat, but whether all these pharmaceutical companies have taken into consideration all these mutations, or then there will be another booster we will have to take. That's the kind of information I hope will be addressed through this talk. And another one is, of course, right now in Canada, the government continues signing contracts with different various pharmaceutical companies, and some are saying Johnson and Johnson is going to be better. Somebody's saying that other one is going to be better. Only time will tell, and it comes back to Chayaji's question. Which one, and second dose, shall we delay, not delay, all those kind of questions.
Great, thank you so much, Ashish. So we are live today on YouTube as well as Facebook. I can see we have viewers on YouTube as well as Facebook right now. So I would invite them to type in their questions as well as we go along. This is usually a hot topic, and this is the third talk, in fact, on vaccination. So let's get back to the. So all of you know about me by now. So I'll skip the disclaimer for today. Get straight to the talk. Today's talk, I will experiment with something new. I will use my whiteboard on my Google Meet to try to demonstrate certain things to you. So it might look a bit like a lecture, but I hope you'll enjoy it. So I'm going to share my screen. Here we go. So you should be able to see my screen by now. Great. So the first, we'll talk about all the vaccines today, and I will also try my hand on my whiteboard. I'm not a very good drawer, but I'll try to demonstrate some things by drawings as well today. This should be quite exciting. The Pfizer vaccine, it's a mRNA vaccine, 2 doses 3 weeks apart, made in the US, 95% efficacy to prevent symptomatic illness. Side effects, there are around 11 cases of severe allergy, what we call anaphylaxis, in every million doses. So that is coming down to roughly 1 in every 100,000. It's important to note that this is quite an acceptable side effect profile for most vaccines. There are several vaccines which have a higher side effect profile than this, which we license and use quite readily and quite happily. Logistics is difficult with that 70°C cold chain requirement, minus 70 degrees, and they are trying to they are trying to see if they can come up with a freeze-dried version, which might be easier to transport, but that would require licensing and testing, and that will take a good few months. It's being produced in the US and Europe. It's highly efficacious. The biggest downside I see is that it's expensive, about $20 a shot. and the allergies, particularly any patients who have allergy to PEG. PEG is something we commonly use. Patients who've had a colonoscopy will have had PEG beforehand to clean themselves out. It's called polyethylene glycol. It's often used for constipation and also used for cleaning out the gut. Patients who have PEG can be allergic to this vaccine because the nanoparticle contains PEG within it. The next vaccine to talk about is Moderna. Of course, these are the two licensed vaccines at this time in North America, in the US and Canada. Moderna is also an mRNA vaccine. 2 doses, 4 weeks apart, made in the US. I like to point out that the several CEOs and founders of Moderna are Canada based, and most of them did their masters in McGill University near Toronto, and then did their PhDs at MIT in Boston, and then set up Moderna in Boston. So it's quite a nice happy Canadian story there as well, but very similar to Elon Musk and so many other people who do their secondary education and university in Canada and then move on to Stanford or MIT to do their PhDs. It's that brain dead drain kind of situation, which is completely understandable. I mean, Canada takes the best from the world, and then usually the best of those are taken up by the US. Minimal side effects. Minus 30 Celsius storage, so it's a bit easier than Pfizer. For that reason, in the community, in nursing homes, for example, in Canada, Moderna vaccine is being used in nursing homes, while the Pfizer vaccine is being used in hospitals. Being made in US and Europe, best advantage is it's efficacious. Con is that it's the most expensive at $30 of one injection, which means 2 injections cost $60. So Moderna is actually the most expensive vaccine. So here I'll get into my little presentation mode. I would like to try my whiteboard and see how it works. Let's see. Let me know if it works out, so I can make a little drawing. We will do a bit of let's say basic high school science today. So let me know if it are you able to see my whiteboard, all of you? I'm able to see it. Not sure if this is visible on the Meet. I've got a whiteboard here. Ashish, are you able to see my whiteboard? No, I think this needs to open.
Yes, we are.
You are? Oh, perfect. Okay.
Yes.
Brilliant. Okay, good. So that's great. Although I'm not sure if the whiteboard is coming up on Meet. It doesn't look like it is. So I'll have to just present again, I think.
It's coming on Meet.
Yes, it's coming on Meet, but it's not broadcasting on YouTube, and it's not broadcasting on YouTube, which is not a problem. That can easily be fixed. If you just give me 1 minute, I will broadcast it on, because this is really interesting. This is something I'm really looking forward to. Seemed to be working yesterday, not working today. Anyway, we'll skip that part, I guess. But what I'll do, I'll just try to explain it without a diagram. Okie dokes. So let's go back to the thing. Right, so what happens is with mRNA vaccines, if I just, for those it's been some time since they did high school science, I'll try to explain what a virus is first. A virus essentially is some genetic material. Usually it's an mRNA, it's either an RNA or a DNA virus. And generally it will be surrounded by some kind of coat, a membrane of some kind. Some of them are without membranes, but most of them do have membranes. So what happens there? And then if you talk about the cells in your body, you have the outer membrane, and then you have the nucleus, the core. You can almost describe it like the brain of the cell, which contains your DNA. Of course, we contain DNA. Viruses contain either DNA or they contain RNA. This particular virus is an RNA virus. Also, we would describe as a positive sensed RNA virus, which means once it goes into our cells, it doesn't need any kind of further steps. It can just straight away get into the ribosomes and produce its proteins. Anyways. And then in the cytoplasm, on the outside of the cell, you have something called ribosomes. Ribosomes pick up RNAs and produce proteins. Proteins do various functions in our body. Antibodies are proteins. Hemoglobin is the globin is a protein. Muscles are made up of protein. So proteins, they carry out various functions in the body. And so that is just a basic structure of a virus and a basic structure of our human cell. So what does an mRNA vaccine do? An mRNA vaccine is basically a messenger RNA, which is essentially like a telegram or a code, which can get into a ribosome in the cytoplasm of your cell and produce a protein that you want to produce. In this case, the protein that has been chosen to be produced is what we describe as the pre-fusion spike. The pre-fusion spike of the coronavirus. So what Moderna and Pfizer have done, and this is brand new technology, so far they had one similar vaccine for horses in veterinary medicine, none for human beings, that they created an mRNA using the genetic sequence. They put a few stabilizing factors in it. Then they covered it up with a lipid nanoparticle. They inject that into your body. Your cells take it up. The mRNA gets into the cytoplasm of your cells, interacts with the ribosomes. The spikes are produced. The spikes, the spiky bits of the coronavirus, not the entire coronavirus, of course, just the spikes. Those are then expressed on the surface of that cell. Your immune system, which is like your security guard, sees these spikes, and then your body produces antibodies against those spikes. So that's how a mRNA vaccine works. They're essentially giving you the genetic code to produce the spike yourself. So you actually do all the hard work, and which is the reason that these were the quickest vaccines to be produced. Because if you think about it, there is not a lot of lab work. If you compare to Bharat Biotech, for example, they have to have tons and tons of live virus which they have to inactivate and work with. In comparison, here everything is being done in a lab. It's almost like digital recording versus analog recording. If you're recording a song, it's like Bharat Biotech is like recording a song with 100 instrumentalists and violins and guitars, and Moderna and Pfizer is like making a song on your computer. It's completely digitalized. The hard work has been taken out. It's your body which does all the hard work. They just give you the code of how to produce a spike. So it's a very smart vaccine. And a lot of people are seeing this as the future of vaccines, because they're appearing to be very safe. All the side effects so far have really been related to the peg within the nanoparticle, which is a covering. Because you have to have some covering for that mRNA. If that mRNA went in without a covering, it would get destroyed straight away. It would never be able to get into the cell. To get into that cell, it needs that fatty covering, which we describe as a lipid nanoparticle. So that is a quick description of mRNA vaccines. Now, as we go along and describe adenovirus vector vaccines, and then inactivated vaccines, you will appreciate the description a bit more. So let's get to the next vaccine. Oxford AstraZeneca vaccine. So this is described as a DNA recombinant monkey adenovirus vector vaccine. It's 2 doses 4 weeks apart, well, 4 to 12 weeks apart. There's been a lot of talk about them wanting to space out the dose, and there has been some research suggesting that that may well be a good thing to do. There's recently been a trial which showed that if you space out the dose even further, then you can get a better immunological response. So that's not necessarily a bad thing. Of course, it's been made in the UK. Overall efficacy for the licensed dose, which is 2 full doses, is 62%. There were some neurological side effects reported: transverse myelitis, Bell's palsy, 1 case of encephalitis, but all of these did resolve, and they were also said to be equal in incidence to the normal population. So AstraZeneca did not feel that these were related to the vaccine. It is normal fridge storage, is being produced globally, including in India. The pros are mainly the cost and logistics are good. Cons, the efficacy is, among all the main vaccines, it appears to be a bit less at 62%. I certainly have heard a bit more about side effects with this vaccine than I have with other vaccines. Some postulate it may be related to the fact that the vector is a monkey adenovirus, and the body kind of reacts more severely sometimes when you essentially instill that kind of vector. And of course, in Germany, they haven't licensed it above the age of 65 because they say there was insufficient data to support vaccination age above 65, which I personally disagree with. There's no reason why you should not vaccinate the elderly, because those are really specifically the patients who need to be vaccinated. To me, it makes no sense. Anyways, so that is the Oxford vaccine. I would like you to just just pay attention to this, the vector, because interestingly, we have 3 vaccines on today's list: the Oxford vaccine, the Johnson & Johnson vaccine, and the Sputnik vaccine, which are based upon the same platform, but interestingly, they use a different vector. And we will talk about that as well. And we will talk about what a vector is. So Oxford vaccine. The next one is Johnson & Johnson. It just came out with its phase 3 trial results. They have applied for EUA. It's a single dose vaccine, which is the biggest plus point of this vaccine. It was made in the US. Overall efficacy appears to be 66%. Here the variant situation comes in. These later vaccines are now being tested against variants as well. And so in the US it was 72%. In South America, where you have the Brazilian variant, efficacy was 66%. In South Africa, it appears to be about 57%. They actually said there were more side effects in the placebo arm than the vaccine arm, which is quite extraordinary. Normal fridge temperature, being produced in the US and Europe. The biggest pro is a single dose. Con, maybe it's relatively low efficacy. However, you have to take it with a grain of salt. This was done in multiple countries and was exposed to variants, which reduced the overall number of efficacy of this vaccine. Bear in mind the vector. It's not a monkey adenovirus vector. It's the human adenovirus, number 26. The third adenovirus vector vaccine that we'll talk about is a Sputnik vaccine made by the Russians. And here, this is very interesting. They decided on a different strategy, and I will explain to you why they went on that strategy. They decided to use 2 adenoviruses on the 2 doses. So first, you will get the adenovirus vector 26, and next, you will get adenovirus number 5. And why the Russian vaccine decided to do that is very interesting. And when I explain the vaccine technology to you, you will then understand why they would have thought about using 2 different vectors. 2 doses 3 weeks apart, which I like. This is similar to the Pfizer vaccine, so you actually get your vaccination a bit quicker, 1 week quicker. Made in Russia. efficacy in the Russian trial, 91.6%, very impressive. Uh, However, this was only done in Russia. They are doing trials in Dubai, UAE, India, and other countries, and we will look forward to those results coming out. But for the Russian population, they have very good efficacy. Mild side effects, it can be stored in a normal fridge if you get the freeze-dried version. Otherwise, the liquid version needs a freezer. It's being made in Russia. It's cheap. The biggest con I would say is that the only data we have right now is Russian data, and some people would be a bit skeptical and not be able to trust that data very much. Would look forward to data coming out from other parts of the world. Particularly, it would be nice to know if it is efficacious against the variants or not. So let's quickly talk about how adenovirus vector works. So this is also very interesting. Before this, the only adenovirus vector vaccine was for Ebola virus, which was interestingly produced in Canada. It was a Canadian university which first invented that vaccine, and then it was first produced by Merck, in a New York-based American company. So what you do in that is, when we talk about RNA and DNA, every RNA has a complementary DNA and a DNA has a complementary RNA. So you get adenovirus, which is actually a DNA virus. First, you have to figure out the complementary DNA of the spike protein RNA. So you know what the spike protein RNA is, spike protein of the coronavirus. You figure out the complementary DNA. Then you take the adenovirus. Now in the case of Oxford, it's a monkey adenovirus. In case of Sputnik, it's two different adenoviruses. And in the case of the Johnson, it's one human adenovirus. Then you do some genetic manipulation with the adenovirus. You essentially cut off parts of its DNA. Particularly, you cut off the parts which allow it to replicate. So that makes the virus a non-replicating virus, which means once it gets into your body, it cannot multiply. That's why it cannot give you an infection. So you cut out those parts, but you paste in the genetic sequence of the spike protein of the coronavirus. So that's what you do. So you take an adenovirus, it's a live virus, you cut out its replication, you paste in it's the complementary sequence for the coronavirus spike protein RNA sequence. There are also certain types of parts of the DNA which can be carcinogenic or which can cause cancer, because it can cause excessive multiplication of cells, and those parts are also cut out during that process. Then you introduce that live virus into the human body. That live virus infects your cell. The DNA of that virus then goes into the nucleus of your cell, produces mRNAs. mRNAs then come out into the cytoplasm, interact with the ribosome. Then the protein the spike protein is produced, expressed on the outside, and your immune cells recognize it and produce an antibody. So it's a slightly more long-winded way of doing it. With the mRNA vaccine, the mRNA goes into your cytoplasm, ribosomes, protein produced, antibody response. With this, it's a slightly more long-winded way of doing it. You have to do all the DNA manipulation. You have to manipulate that live virus. The virus is what infects your cell instead of a nanoparticle. It infects your cell, goes into your nucleus, produces mRNA, ribosome, protein, immune response. So it's a long-winded way of doing it. Now this part of which adenovirus they used is very interesting, because these vaccines can become less efficacious if you have immunity against that adenovirus. So if you have already these adenoviruses cause normally mild illnesses, common colds. If you've had an adenovirus common cold before and you already have antibodies against that adenovirus, it can neutralize that adenovirus before it can even infect your cell. And there are different types of antibodies we have. We have IgM, IgG, IgE antibodies. IgE antibodies are the ones which are on the mucosal cells, and they're very early Now, of course, these vaccines are not being given through nasal spray; they're being given into injections. But if you already have antibodies against the adenovirus, you may make that vaccine less effective because it can get into your cell. So to counter that, what Oxford did was that's why they used the monkey adenovirus, because they figured this is a virus which affects monkeys, so human beings will not have natural immunity against it. And that's why the Sputnik used two different human adenoviruses, because first you get the first shot and you will produce some antibodies against that adenovirus as well. The question is, when you get the second shot, will you just neutralize that adenovirus? That's why they gave you a different adenovirus for the second shot, and it appears to have worked. Looking at least the Russian data, it appears that worked quite well. Of course, one wonders why the Oxford vaccine didn't work so well. I mean, if it's a chimpanzee or monkey adenovirus, humans should have no natural immunity against it. But one also wonders why the side effect profile has been slightly higher with the Oxford vaccine, because of the fact that it's a zoonotic virus they're using, a non-human virus that they're using with that. So that's the adenovirus vector vaccines. Next, let's move to the newest kid on the block, Novavax. Now, Novavax is very interesting, and I will, disclaimer, this is my opinion, but this is my favorite vaccine. And I'll explain to you why it is. It's 2 doses, 3 weeks apart. It's a protein subunit vaccine. It is developed in the US. Overall efficacy was 89%. However, against the original strain, it was 96%. So you could actually describe this vaccine as a topper. If you remember, Moderna 94%, Pfizer 95%. Against the original strain, this was 96%. 86% versus UK variant, 60% against South African variant. Mild side effects, normal fridge. It's going to be produced globally, including in Canada and in India as well. It's a time-tested platform. This is the same platform that we use for our shingles vaccine, Shingrix, and also the hepatitis B vaccine, Engerix. I cannot see any disadvantages of this particular vaccine, Novavax. Now, a lot of people have been criticizing the Canadian government, and a lot of people have been criticizing Justin Trudeau for being very late at deciding which vaccine would be produced in Canada. And I think he got lucky. Canada had signed a deal with CanSino, which was a China and Canada collaboration and they had made a factory in Canada for producing the CanSino vaccine. That CanSino vaccine was a human adenovirus vector vaccine. By luck or good fortune, Canada and China started having some issues, some political issues, political grumblings. China refused to cooperate, did not send any doses for the phase 3 trial to Canada, refused to export the vaccine, and the whole thing broke down. That was probably very good luck for the Canadians, because the vaccine actually turned out to be quite a poor vaccine, had quite a high side effect rate, was not very effective, and now China is selling this vaccine to Pakistan and other countries. But so now the Canadians have signed a deal where they will change that CanSino factory to produce the Novavax vaccine, which just by a stroke of luck now appears to be the best vaccine, in my opinion. I would like to very strongly explain here, though, all these vaccines are really quite safe compared to a lot of other vaccines we have out there. These are really quite safe. If you get a chance to get a vaccine, go ahead and get it. It doesn't matter which vaccine you're offered, because it's much better to have a vaccine than not have a vaccine. I would not want anyone to wait for this vaccine and for that reason not get a vaccine which is offered to you, because any vaccine is better than no vaccine, and you don't want to end up contracting COVID-19 while you're waiting for a good vaccine like this one. But I really like this vaccine, and let me explain to you why I like this vaccine. So I'll just move back to explaining a protein subunit vaccine. A protein subunit vaccine, what they do is, first they do DNA recombination or DNA manipulation of a virus. In this case, it's something called a bacillovirus. So they do the usual steps that you would do. They inactivate it to the extent that it cannot replicate. They make it non-replicating. They take away any carcinogenic potential. They put in the DNA sequence, which would give you RNA to produce the spike protein. However, what they then do with this bacillovirus is they infect it into moths, larva moths. So these are moths which were about to produce a butterfly. They infect the moths with that virus, and the whole function which would have happened inside your human body with the adenovirus vaccine actually happens inside the moth. So inside the moth, that virus gets into the DNA. The DNA produces messenger RNA. RNA interacts with the ribosome, produces proteins. Proteins are expressed on the surface. The antibody response takes place. All of that happens not inside you, that happens inside that moth, that caterpillar. Then what they do, the spike proteins which were produced, they harvest those spike proteins from that moth. And so it's a bit like producing honey. Just like you have bees and then they produce honey and you harvest the honey, they actually do the whole process inside another organism, in this case a butterfly, and then they harvest the spike protein that we really wanted, the end product. Then they coat that spike protein within a particle and they give it to you as a vaccine. The interesting thing is, it will not infect any cells. Straight away, while it goes into your body, it is picked up by your immune cells, macrophages, dendritic cells, fibroblasts, and antibodies produced. There is no stage of infecting your cells, going into cytoplasm, going into nucleus, producing stuff. It's already produced. All that work has been done outside your body. So that spike protein has been produced outside, externally, and just given within a particle. So if you think about it, this is the vaccine which virtually does hardly anything to your body. You're just given a spike protein within a particle. We call this a virus-like particle, VLP. It has been designed to mimic a virus. So it's been given a shape and size which is very similar to virus. So your body just picks it up like it's a viral infection and produces antibodies. But of course, it only contains the spike protein. It does not contain any of the other. It has no RNA. It has no DNA. It has nothing which is living. So that's the Novavax protein. Novavax protein subunit vaccine. And finally, we will talk about Bharat Biotech. And being Indian, I think we can be very proud of the Bharat Biotech vaccine. And I will tell you something. Just 3 days ago, the stock price of Bharat Biotech really went up because they sold. They did a deal with the US for selling Bharat Biotech vaccine in the US. It's with a company called Oxygen. And just yesterday, I was listening to JAMA, and JAMA is a top-level medical establishment in the US, and they were talking about inactivated vaccines like Bharat Biotech, and they were saying, considering that we are now getting so many variants, should we be considering inactivated vaccines? And the reason for that is because an inactivated vaccine contains the whole virus. It doesn't contain just the spike protein. Before this, all the other vaccines we talked about contained, one way or the other, it ended up with giving you the spike protein, which is the spikes on the coronavirus. Not with Bharat Biotech. What happened with Bharat Biotech? Let me explain, but let's first talk about the vaccine. It's 2 doses, 2 weeks apart, produced in India. We don't know the efficacy. The phase 3 trials have not been done. Appears to have minimal side effects so far. About 25,000 people have been enlisted within the trial. Normal fridge storage. This is the oldest and safest platform that we know. Children from age of 2, the vaccines they receive, like polio vaccine and tetanus vaccine, these are all inactivated vaccines. We don't know what the efficacy is. Typically, this tends to be a very mild platform. That's why the children's vaccine, you have to give it at 2 months, 3 months, 4 months, because these tend to be very mild vaccines, but they tend to require one adjuvant, like alum-like substance, to try to stimulate the immune system, and they tend to require multiple boosters as well. So that's the pro and con. The biggest pro is its con as well. The biggest pro is it's a mild vaccine, so very less side effects, but the con is also it often requires multiple boosters because it is so mild. It is not as immunogenic typically as the other vaccines we're talking about here. So how do you produce inactivated vaccine? So first you have to grow a lot of virus. So in the National Institute of Virology in Pune, and in the they would have cultured a lot of coronavirus. They would have probably used a strain which is prevalent in India at that time, and then they would in the lab inactivate it. How do they do that? They then mix in a chemical which goes into the nucleus, or rather virus, or it doesn't have a nucleus, goes into the virus and inactivates the genetic material. It produces blocks within the genetic material of that virus. Essentially it kills that virus. However, what happens is that virus still has a body. It still has a membrane, it still has the spikes, it still has the nucleocapsid protein, but the inside, the RNA has been destroyed. So this is essentially, they kill the virus and turn it into, you could describe it like a dead body of the virus. And then they collect the dead body of that whole virus and inject it into you. So the good thing about this kind of vaccine, it is not just exposing your body to one part of the vaccine, of the virus, which is the spike. It exposes your body to the entire body of the virus, the membranes, the coverings, everything. So actually you wouldn't just produce 1 or 2 antibodies, you would produce thousands of antibodies attacking different parts of that virus. So that's how this vaccine works. The reason that most countries try to avoid doing this is because it's very labor intensive. Producing a vaccine like this is labor intensive. Producing an mRNA vaccine, you can basically produce it on a computer. Once you have the genetic sequence, and the genetic sequence was released on the 10th of January on Twitter by the Chinese authorities, the mRNA vaccines could get working on that straight away. And that's why they were the first to come out. Pfizer was licensed early December. While to make an inactivated virus, you have to have tons and tons of live virus. You have to have a grade 3 manufacturing facility. It is the one for Bharat Biotech is one of a kind. In fact, I listened to the CEO of Bharat Biotech saying that even in the US they don't have a grade 3 manufacturing facility for live viruses. So even in the US they cannot produce this vaccine. They can test it. They have testing facilities. They have labs, but they do not have production facilities. And I've even heard that the workers at Bharat Biotech had to stay away from home. They had to live within the factory for up to 6 months at a time because they were working with live viruses. And so the risk was very high. And that's why most countries do not want to produce these vaccines. Only 2 countries that I know of, China and India, have produced these. Sinovac from China, and India have produced this vaccine. However, I'm really looking forward to efficacy data coming out for this vaccine, but I expect side effect profile to be very low because there are virtually no moving parts with this vaccine. It is very simple. You take the live virus, you kill the insides, the RNA, and you just present the dead body to the body. That's basically it. And efficacy data might be on the lower side. It may be between 50 to 60%. That should come as no surprise. But I have to say something about efficacy data as well. The really important number isn't efficacy to prevent serious illness or symptomatic illness. It should be efficacy to prevent hospitalization and death. And so far, all the vaccines we've talked about so far, all of them have 100% efficacy. And so to say that Oxford has 62% or Johnson is 66% or Novavax is 96% is probably a bit unfair because they are all 100%. No, so far more than 10 million doses of these vaccines have been given, and there has not been a single case of a person who received both doses of the vaccine of course Johnson has a single dose, but who received both doses of the vaccine, waited 2 weeks, which is when the maximum immune response has happened, and then died of COVID-19. Out of 10 million people, not a single case. And I can assure you, if you get a single case, it will be in the headlines. You know, the fact that 15,000 people yesterday died of COVID-19 is not in the headlines, but if one person receives a vaccine full dose and dies of COVID-19, it will be in the headlines. I can assure you of that. But so far, no headlines of that sort. So to reduce death or prevent death from COVID-19, all of these vaccines are 100%. And so we are very lucky as a human race. We really have to, salute the scientists and people who have gone working in the background producing these vaccines. This is a real scientific breakthrough. So the average time to produce a vaccine used to be about 10 years, and now it's been compressed to 11 months, which is incredible. So many people that I follow on social media, they have not taken a single day off. They've been working 7 days a week flat out for the last 12 months to produce these vaccines, and we really have to appreciate the enormous effort that these people have made, and they will now be responsible for saving millions and millions of lives. So it's just an extraordinary story, and I'm sure a lot of the Nobel Prizes in medicine and biochemistry, I would imagine immunology would go to these people who have come up with these incredible breakthroughs in vaccine development. And because these are all 100% effective, I don't feel that we need anyone who's offered a vaccine should not take the vaccine. I think if you are offered the vaccine, you should go ahead and take the vaccine. So I will ask all of you to quickly just ask me the question again that you asked me one by one, and then we will answer these questions one by one. So I'll start with my parents. I remember the question. The question was about the efficacy in older people and also about Bharat Biotech. But Daddy, Mummy, if you could just give us your views on today's talk, and also just repeat your question, please. Thank you.
Yes, well, the points are already clear that once the vaccine is offered to us, then we should go for it. That's it. Because the benefits are much more than the risk, which is almost nil. That's it. Thank you.
Thank you so much for very informative talk. You helped everyone. You helped us so much, and we are gaining so much from you. You are doing a great job. Thank you, Dr. Sapan Singh. God bless you.
Thank you.
Thank you, Sapan. This is great information, and this is like pros and cons of all the vaccinations provided in one lecture. So that is great. Yeah. Maybe you should also document it, like whatever. I mean, in your presentation, it's obviously there, so we can always refer it, on YouTube. So that's amazing. Thank you so much.
Bhavika, do you have any questions? So when we started off,
So my question was that as a doctor, you would know what vaccine I'm going to get if I come to your office, and would you say this is what you're getting, and would we have a choice? That was my question. And the second part was, I think that was the main point was, would I know what vaccine I'll be getting? It's a lot to learn.
Seema ji and Ashish, do you want to just have some comments before I start answering all the questions about what you thought about the talk, and just repeat your question for me as well. So Ashish, perhaps you can go next.
Thank you, Dr. Sapan, once again, very informative. It was a great, presentation. Also, we have forgotten everything about viruses, and so it was a good refresher also. And my question was various strains coming out, and how long the vaccines would be effective. Some are saying they would be for 6 months, a booster will be required in 12 months. And with a lot of variants coming, would this cover, or then we will have to take boosters later on? Thank you.
Hello, Sapan. Yeah, it was a great talk. And as you made clear about the pros and cons of each and every vaccine, I just know like which we have to take. But as Chayaji was asking, do we have a choice here to take the vaccine? That is what is, yeah. And the long term, right, long term side effects of that, yeah, that is what we have to make clear. Yeah.
Um, so even I have a question. I think it's something I've already discussed with Sapin, personally, but then just like curious about how things will go about for breastfeeding mothers and pregnant women with vaccination, because I know they've not been able to test it on pregnant women right now. So, what is the future we see, for this category of population?
Great, thank you everyone. So let me just answer Bhavika's question first. I'll just go in random order. So Bhavika's question is about vaccination in pregnant and breastfeeding women. So there is differing guidance on this. So CDC and other authorities have differing guidance on this. But because there is no trial data, we cannot be definitive with anything. However, generally speaking, anything which does not have a live component is generally speaking safe in pregnancy. So although the adenovirus vector vaccines, which among this group includes Oxford, Johnson, and Sputnik, have a live component, although it has been inactivated to be non-replicating, I would still advise caution for pregnant and breastfeeding women. I would suggest they should avoid it. So in India, they should probably take Bharat Biotech because it's an inactivated vaccine. In North America, if you had a choice between mRNA vaccines and Novavax, I would go for Novavax. However, mRNA is also it has no live components really, so you could go for that as well. Chayaji's question and Seema's question about choice, it's very unlikely that any of us will be given any choice. I mean, I'm a physician. I haven't gotten a call here in Canada yet because of for the vaccine, because there was such a shortage. They divided doctors into hospital-based and community-based. We come under community-based doctors, hence we are not being offered the vaccine yet. And I guess it'll just be a case of as soon as you can get your hold on one, go ahead and take it. I mean, generally speaking, these are all very safe vaccines, and no one should really have any qualms about it. Of course, if you get into a situation, if you're in a very low risk group, as an example, I would say, several of you here are young people, so you're in low risk groups. It is possible by the time your turn comes that you have a few choices. And then at that time, we'll have to evaluate. Of course, the situation also changes according to the strains. As we saw, the vaccine which came out later, like Johnson and Novavax, have been tested against strains as well, while the ones which came out earlier have not been tested against the strains. Although they have done neutralization assays within the lab, and Pfizer came out with a statement saying that it appears that they neutralized the South African strain. I suspect they will all be very similar with their efficacy against the strains. The reason for that is because they're actually all using the same technology, the prefusion spike. There was a company in Texas and one in Vancouver. You might remember the one in Vancouver which produced a monoclonal antibody, which came bamlanivimab. It's called Accelera. It's a Vancouver, Canada-based company which came up with the sequence for a prefusion spike, which they have used for monoclonal antibodies. And most of these leading companies or vaccine producers are using that same formula. So even though they're giving you that spike protein in a different form, in a different way, so mRNA, either injecting mRNA into you, injecting it through an adenovirus vector, or producing the spike outside your body in a caterpillar and then giving you the protein directly, it is actually the same formula at the end. So if one vaccine is becoming less efficacious for it, I suspect all of them will start becoming less efficacious. Saying that, the good thing here is that most of these vaccines within 6 to 8 weeks can adjust the formula. They can change this genetic sequence that they are, the code that they're putting in, and they can make a vaccine which is better for the variants. So I don't feel too negatively about it. I feel that even though the variants is becoming a bigger problem, the science is there to be able to counteract it. Of course, the one vaccine which does not follow that formula is the Bharat Biotech vaccine, because it's not just giving you a spike, it's giving you the whole virus. Let's talk about, Daddy had a question about using vaccines in older people. Now, what the German authorities said was that the Oxford vaccine is I think it's about 11,000 people, or rather it was maybe 11% of the trial was done in people above 65. They felt that they hadn't done it in enough people above the age of 65, hence they did not license it. I think that was an opportunity missed. I don't believe in that. It's going to work in most age groups. Now Germans are also producing the Pfizer vaccine. And of course, BioNTech is a German company. So I suspect there might be a bit of a political decision there as well, because they wanted to use their Pfizer vaccine, and it seems reasonable. I mean, Pfizer vaccine has a 95% efficacy rate. Oxford vaccine is a 62% efficacy rate, and Pfizer is their own, BioNTech is their own German company, so they decided not to license it. The Germans don't get along with the British, generally speaking anyways. They've had 2 wars. Britain just came out of the European Union, so I think it's a combination of politics and all of that, and they're kind of rubbing in the fact, look, our vaccine is 95%, your vaccine is 62%, so we won't even license you. So I think it is just that kind of a bit of a envious kind of relationship that they have between the two countries. I think that's a bit of that going in. So Seemaji,
Sapan,
yes.
sorry, I am not too sure if you're done with all the questions, but I also have one more question to the answers that you have given. So thank you so much for the answer first. But my question is, let's say, because as of now, the status quo is that they're not giving vaccination to pregnant women and breastfeeding mothers. So if a person who's vaccinated, so let's say if I have 5 people in my family, everybody's vaccinated but myself, can they carry the virus and still infect me? So would the family members still need to be as careful as they were before the vaccination, just because there's somebody in the house without a vaccination? Or because they have the vaccination, they will never get infected or they'll never carry the virus?
Yeah, so the trials are being done for that. Of course, everyone is trying to be very cautious with this question. And because we haven't done the trials, we are not able to give the answer. The most recent trial which came out was for the Oxford vaccine, and they did appear to reduce transmission by over 75%. So common sense says that if you're less likely to get the infection, you're less likely to transmit the infection. So, and the Oxford trial did seem to suggest that, and I would be very surprised if that was not reproduced for other vaccines as well. So personally, I feel that if 2 people have had the vaccine and they want to meet and not wear masks indoors, of course they've allowed the full 6 weeks to pass for the vaccine to have full efficacy, and let's presume it's a good vaccine, it's a good efficacious vaccine. Personally, this is my personal opinion, I would have no problems meeting that person indoors, because you have to remember, all these vaccines are still effective for 100% for preventing infection, for preventing death. So you have to take some risk, in the sense that even if you accept the risk that there is a very small chance I might get the infection, the chances that I'd end up hospitalized and dying because of COVID-19 is virtually 0%, and hasn't happened yet after 10 million doses of the vaccine. So, I would be okay with it. And personally as well, I don't see COVID-19 as something that I want to completely avoid. I don't see it that way. I see COVID-19 as something which I don't want to get hospitalized with. That's my target.
Right.
So if I can achieve my target of not getting hospitalized with COVID-19, I think that's a good enough target. I think all of us are getting a bit like, it's a bit like 12th grade board exams, who got 95%, who got 62%, you flunked, you passed. It's not really like that. I think if we all start becoming a bit more pragmatic with the whole situation and thinking, this is, we're getting millions of cases now per day. It is not possible really to eradicate it. If we can just reduce this condition to something which gives you like a cough and cold, gives you a flu-like illness, and you don't end up hospitalized, that's as good as it gets, and that's a very big achievement. And at this time, it appears like all of these vaccines are able to give you that result, which is great, which is exactly what we need.
Okay, so then it's just that everybody should still be cautious of the fact that this virus exists even after the vaccination, and just the effects of of the virus can be reduced because of these vaccinations. I think that's the conclusion I gain out of it.
I think what's happening also, Bhavika, is the strain situation. In South Africa, they've had lots of reinfection. People who had COVID-19 in the early phase in March, April are now getting it again because of the strains.
Right.
And so,
Hmm.
and the strains are also escaping a lot of vaccines. So about 50% to 60% in the Novavax and the Johnson trials. And so Novavax did 96% against the original strain, and in the 50s, in fact, was 60% against the mutant strain. So the strain situation just does change the dynamics slightly. Now we can't say to people anymore that you won't get reinfected. You could well get reinfected. However, even now, these vaccines, even against the newer strains, have still protected 100% from dying of COVID-19. So that amount
Oh,
of
that's great.
yeah, so far that amount of protection is still there. So, and I'm sure these companies will keep changing the sequence probably every 3 to 6 months. They will keep changing the code of the DNA, of the RNA within their vaccine, to keep on top of the various strains which keep forming. And this is not nothing new. This is what we do every year for the flu vaccine. Every year for the flu vaccine, we
Right.
have a year-round surveillance program. There is genomic studies being done for the flu vaccine, and every year we come up with a new flu vaccine, which is a new vaccine to account for all the mutations and all the strains which have developed, all the variations which have developed. And so I suspect the same
Mhm.
situation will be happening with COVID-19 as well.
Sounds great. Thank you, Sapan. This is very thorough. Uh, Thank you.
I think Ashish was asking about strains, so I'm going to just cover the strains. So, of course, we have a few strains which are, or rather, we shouldn't call them strains, we should call them variants. We have a few variants of concern. You have the UK based. Now, a lot of people get, a bit iffy about using the word UK or South Africa or Brazil, because it seems like, we didn't use Wuhan or China. Now, why are we using these countries? And it's a bit unfair for them, because in a lot of cases the strain probably didn't originate in those countries. It was probably picked up in those countries because they do a lot of genomic surveillance, while other countries are not doing genomic surveillance, and for that reason they didn't pick it up. In any case, but it's much easier than remembering, B1552 or B951. You can't remember those numbers, so it's easier just to call them UK variant, Brazil variant, South African variant. The UK variant, I am concerned about this variant. I have to be honest, because recently there was this news article from this nursing home in Ontario where the UK variant got into the nursing home. It infected about 127 people in that nursing home, and about 66 people died. So that was over a 50% case fatality rate in Ontario because of the UK variant. So the so far we haven't seen, even in older people, that kind of fatality rate. We haven't, even in people in their 70s and 80s, it has been around about 20 to 40% maximum. It seems to peak out at that for the old variants. So just a week ago, Boris Johnson the UK Prime Minister had announced that it appears that not only is the UK variant more transmissible, probably they were saying anything from 30 to 70% more transmissible, it appears to be about 30% more fatal as well, more dangerous as well. And I feel that even as a case study in itself, the fact that it ended up wiping out more than 50% of this one nursing home, it seems to me enough proof that this variant is dangerous. Because so far I haven't heard of another case of more than half of a nursing home dying because of COVID-19. So 66 people in one home, you can imagine, it's devastating for the families and for the home itself. And so I feel it is definitely a concern. The good thing about the UK variant, though, is that it appears to be more susceptible to vaccines. So vaccines do appear to protect against the UK variant. Of course, the big challenge is getting the vaccine. The rollout has been slow, particularly in Canada. The rollout has been very slow because of the issues with procuring or getting these vaccines. Because as more and more countries are licensing them, everyone wants some vaccine. So Pfizer and Moderna, how many countries can they provide to? So that's completely understandable. The South Africa variant seems to be more problematic with regards to with escape from vaccine and escape from monoclonal antibodies. That's a bigger concern for the South Africa variant. So I believe the vaccines, Moderna, etc., they will have to work quicker on the South Africa variant to make their vaccines more effective. Of course, I'll repeat again for the 10th time that when we talk about efficacy, we're talking about efficacy for symptomatic disease. So far, it appears that vaccines are still protecting from severe disease and from death from COVID-19. So that's still good. Brazil variant is a bit of a shoot off of the South African variant and seems to have a similar kind of profile to the South African variant as well. So that's just something about the variants. Ashish, did you want to add to that? Because that was your question.
No, it was very informative once again. But then going forward in future, it's going to be a big challenge for all these pharmaceutical companies. Like every year, we get a new version of this what you call flu shot, and there are around 20, 30, 40 variants. But here we are talking now about 4,000 variants. So like Moderna, scientists will have to sit on computer and find RNAs of all those 4,000. And I think it's going to be a bigger challenge for Covaxin, how they are going to grab all viruses, 4,000 from all over the world. And is it going to be a cocktail of those 4,000 inactivated viruses? So it's going to be a challenge, I think, for these pharmaceutical companies.
Yeah, so two things. One, it's less of a challenge for Covaxin actually for the Bharat Biotech, because you see, when there is a mutation, let's say they say, oh, there are 12 changes on the spike protein or 24 changes on the spike protein, but the rest of the virus is still the same. So as long as, because the Bharat Biotech is covering the entire virus, it will still kill that virus. So it's less of a problem. The problem with these other more advanced vaccines is they're focusing on a very small part of that vaccine, perhaps the receptor binding domain, receptor binding moiety of that virus, and that if that changes significantly, the vaccine becomes less effective. So it's a bigger problem for them. Also, I would talk about the strains. Of course, there are many strains, but you have to match the genomic data with the epidemiological data. So for example, in South Africa, 95% of the positive cases are now the South African variant. Also in the UK, now the dominant strain has become the UK variant. So what happens with the mutation is it can affect the virus in two ways. It can either make it more effective or less effective. So there are many variations in the virus which can actually make the virus less effective as well. So, and the only way you can find out, through the survival of fittest test of which variation is actually giving that virus an advantage is essentially epidemiological data. When you start seeing that a certain variant is becoming more dominant because it seems to be spreading faster or any other reason, that's when the vaccine companies would start having to think about incorporating that mutation into their code. And yesterday when I was listening to JAMA, they were talking about mixing vaccines. They were saying, would mixing vaccines help? Would it be helpful to give like a Pfizer vaccine for the first one and then give like an inactivated vaccine like Bharat Biotech for the second one? Because you would then cover more of the virus, while if you give Pfizer followed by Moderna, actually, if you think about it, they're all given the same spike. So what the virologists say is they put all their eggs in the spike basket. All these other vaccine producers, like we say, put your eggs in one basket. They all there's one spike basket which all the vaccine producers put their eggs in that basket. And now they're talking about the other vaccines which are produced by a more difficult method, but actually has a different approach towards the vaccine. But even in India, they will have to keep an eye on the dominant strain. there will be less dominant and more dominant strains, and I'm fairly sure they will be able to culture those strains. I'm sorry. I just pressed a button and came out of the meeting. I'm sure they will be able to culture those strains and adjust their vaccine accordingly. But in Bharat Biotech's case, it is not so much that they have to go for this particular strain, because let's say there are even 10 dominant strains, they will all have a lot of things in common. Their body will be common, their nucleocapsid protein, other proteins will be common. So as long as the virus is attacking one or two parts of that, it will be effective.
So would DNA-based vaccines will have an advantage over RNA-based? No, it's only inactivated whole virus vaccine. They will have an advantage. That's right.
That's correct, because DNA is essentially just a long-winded way of doing the same job which RNA does. So RNA goes into your body, goes into your ribosomes, produces a protein. DNA goes into your cytoplasm, then goes into your nucleus, produces a RNA, then goes into your ribosome and produces a protein. So it's just a long-winded way of doing the same job.
Sapan, I just have a question regarding my question regarding the choices. I'm not a medical person, and I get so confused with all these strange words. So the onuses will be on the patient to be careful, or the doctor will say this may not be suitable for you because you have these underlying condition, or so just what as a doctor in Ontario, what would you decide?
Yeah, it's very unlikely you'll be given a choice, and I would say take whichever vaccine which is given to you. However, for those who are in a low risk group, like younger people, their turn might come in September or something like that, because the target is to vaccinate everyone by September. By that time, we may have a few choices. We may well have 4 or 5 choices, and then I would have to advise those people at that time, because it also depends on a few other factors. Let's say, for example, right now, if you had to choose between 3, let's say a patient is pregnant, as an example, and let's say they had to choose between Pfizer, Moderna, and Novavax, I would recommend Novavax, because Novavax is a protein which is given to you directly. It does not go into cytoplasm, into your nucleus. There's nothing inside your body. All that work has been done inside that butterfly. All that work is already done. They just harvested the protein and gave it to you. So I would recommend the Novavax. However, let's say, for example, Moderna changes their vaccine to incorporate the South Africa strain, but Novavax is late. They haven't done it. They're doing it after a month. Then Moderna will have an advantage. Whichever vaccine has got is updating themselves for the variants quicker will then be a better vaccine at that time. So let's say if for argument's sake, if Bhavika gets the call in September, I will have to advise her just a week or so before which choice might be a better choice at that time, because it depends on so many other factors as well.
Thank you so much.
Seema, what was your question again? I know you were asking about side effects and that kind of thing, but if you just elaborate again, you're muted right now.
Unmute yourself, Seema.
You're mute.
Yeah, like I would like to know the long-term side effects in different age categories for kids, for adults, and for seniors. Long-term, yeah.
Okay, this is a situation which is a bit controversial. So I have to be very careful how I say this. My official line is that these are all safe vaccines, and whichever vaccine you can get, you should get, because it is better than COVID-19. That's my official line. However, now let me just get to my opinion, which is my opinion. It is unofficial. Okay. Long-term side effects. The truth is that obviously we don't really know until you've had it, unless it's been 5 years since the vaccine came out. You don't know what happens after 5 years. Saying that very few side effects come up after 5 years. Most of the side effects do come up fairly soon after vaccines. Also, though, a lot of these vaccines are very new technologies. For example, the mRNA vaccine had only been used in horses before this. The DNA vaccines had only been used for Ebola virus in a small part of Africa before this. So these are new technologies. My feeling is the less that a vaccine does inside your body, the less chances it has of giving you a long-term side effect. So the least is probably Bharat Biotech inactivated vaccine. They just give you the dead body of the virus, the immune response kicks in. Novavax vaccine, they just give you the protein of the spike protein, the immune response kicks in. Those are the simplest vaccines. The next stage would be mRNA vaccines. They go to cytoplasm, ribosome protein. There is the process. Unlikely to cause any side effects. So far, we're not seeing any signals. All the allergies from Pfizer appear to be related to polyethylene glycol, which is in the nanoparticle. But then when you get to the adenovirus vector DNA recombinant vaccines, which is Oxford, Johnson, Sputnik, these are more complex. One, they introduce a live virus into your body. Secondly, the DNA goes into your nucleus. Then the DNA then produces RNA. RNA produces proteins. Proteins produce an immune response. A more complicated way of doing this, introducing a live virus into your body. In the case of Oxford, it is a monkey virus. Question mark, is that related to the neurological complications that we saw because it's a zoonotic infection being introduced? Probably not. AstraZeneca say it's not. Oxford said it's not. They say that the incidence was equal to the normal population. There is no reason why we should not believe them. But my personal view is, if I was given a choice, if I was just given a choice of one vaccine, I would take whichever it is. If I was given a choice of multiple vaccines, I would take the simplest vaccine. I would take the one which does the least according to its mechanism of action, because to me that appears to be what would likely lead to less long-term kind of sequelae. Do we have any more questions? Have I answered all the questions? Because I can now remember all the questions from the very start.
Thank you, Sapan, for the clarification.
Do we have any more questions at all, Daddy, Mummy? Let's do some closing remarks now. And if you have any more questions, you can go ahead and ask any questions. So, Daddy, Mummy, you can start.
No questions, please. Thank you so much for very informative and useful talk. God bless you. Thank you so much, Dr. Sapan.
Well, every time it adds to our information and knowledge. That's it. Otherwise we are laymen, don't know anything about these things. But slowly and gradually we are also becoming aware of these things, medical things. So thank you so much.
Great help. Yeah, great job. Thanks so much.
Thank you, Sapan, for my persistent question. The choices. I'm thinking about it. Thank you for educating us. Even though I don't remember everything, the revision and repetition really helps us to be aware of this dangerous situation. So thank you ever so much. Thank you.
Thank you, Dr. Sapan, once again. You keep on knowledging us, and we are up to speed on this one. Thanks again for your blog and your online sessions also.
Thank you, everyone.
Thank
for
you,
joining
Sapan.
in.
We just love the fact how we can get all the personal answers. Like, I know my question was like very, very categorized for a very specific kind of population, but thank you. Like, it feels great, and there's so much data available on Google. Sometimes it's scary. So at least we know we are getting information from an expert or somebody who's actually thoroughly researched on these topics. So thank you for your these blogs and vlogs. Like, these are very helpful.
Thanks, everyone. Great. So yeah, that's fantastic talk. So I'm glad everyone found it informative, and so we'll meet again. It's same time, same day, Sunday, 10 o'clock Eastern time. We'll meet again. Thank you. Take care. Bye, everyone.
Thank you so much.
Bye.
God bless.